Okay, so once you finish translation, then next you're going to tell students to complete 10 and 11. So for 10, it asks them to compare their sequences to each other. Now, I should tell you. So when they go to actually translate, they're going to use the little beads and they're going to string them on a pipe cleaner. So we should have pipe cleaners out and beads. Please don't let the students take them home. They need to replace the beads back in the bucket, and we need the pipe cleaners back. That's why we're like missing half of our beads. It's because students are taking them home as like a souvenir. Don't let them take them home. Have them unstring them and put them back. All right? So they're stringing them on the beads, and you'll see that this is what it'll end up looking like. Right? Where are we? Yeah. Yeah. So it'll look like this. I don't know if you can see this. Okay. So beads on a string. This is our hemoglobin gene. Okay. So they're going to end up with something like this. Once they've done this, they're then going to compare their sequence to everybody else's. And there's some keys that they're labeled in the drawer back there. We need to have those out just for the TA. You should just have them in your pocket. Okay. And you can go around and you can show yours to them and compare. And inevitably you will have people that have made mistakes and their beads will be different colors, which is fabulous. We want that because then we'll say, that's interesting. This is what it should look like. What happened? And they can like count which bead, and they can go back to their sequence, they can go back to the original DNA, and they can figure out what's wrong. I want them to spend time doing that, figuring out where the mutation was. And as you're walking around the tables, you'll see the different mutations, and that can help you introduce the different mutations, okay? So the mutations you're going to introduce, and I'm going to give you an example. So let's say it was supposed to read what am I doing here? U, and instead I wrote UAC. What is that going to do? Now I would use examples from what your class actually did if you find them. If not, these are good generic examples that you can use. Okay, so if we went from UAU to UAC, let the students look it up, and they'll see that this stands for a tyrosine, and so does this. Now these you probably won't find an example in the class because they won't notice because their bead will be the same. Okay, and this is called a silent mutation. It's silent. It didn't make any difference in the DNA sequence. And we're going to learn, I at least share one in class with spinal muscular atrophy where silent mutations do matter, and we'll talk about that later in lecture. But for the most part, silent mutations don't do anything to the protein. It's the same amino acid, you don't even notice it, okay? But let's say instead of UAU, so it's supposed to be UAU, and I wrote UGU. Okay, so we know that this stands for a tyrosine. What does UGU stand for? So they're going to look it up. UGU stands for cysteine. Uh-oh. So we've just made the wrong word. We've made a mistake. And so we call this a missense. It's the wrong sense. Okay? And a missense, I'd ask them, what do you think that's going to do to a protein? If you stick the wrong bead in there, maybe it has a different charge, maybe it has a different polarity, well, it's going to mess up the tertiary shape of the protein. Badly sometimes. Sometimes not so badly, sometimes really badly. So it's a bad mutation. And then let's say instead of, let's see, let me look at my other example. Instead of UAU, I wrote UAA. Okay, so this is a tyrosine. This is a stop. It doesn't make any sense at all. There's no word there. So it is a nonsense mutation. And nonsense mutations truncate proteins. So you're supposed to be this long and now it's this long. And you might actually see some around that end up with that where their protein is like 3 beads long. That's a nonsense mutation. Those are very bad. Okay? And then the other thing that's probably the most common mistake we see with students is that they cut their intron exon like 1 letter too short or 1 letter too long. By the way, I just want to mention this in here. The introns are not necessarily multiples of 3. So if students are trying to read the whole thing, introns included, they're going to get all screwed up. Okay? Anyway, so let's say they added a couple letters. So they were supposed to write, UAU, but instead they wrote UAAU. Right? Now in that case, it's actually going to result in a stop. But let's say they just put an extra letter in there. Let's do a C. Okay? That's going to screw up the whole downstream protein. See, this is called an insertion. And then the other one is that instead of writing UAU, we just write UU. And then we continue on with our strand. That's a deletion. Both of these are really bad. These together are called frame shift mutations. Oops. Because they shift the reading frame, they screw up the whole protein. These ones are called base pair substitutions. Both can be bad, but the frame shift is particularly bad because it screws up everything downstream from them. All right? So, and this is another thing to emphasize. When they were making these mistakes, they were making them in the mRNA copy. These mistakes do happen in the mRNA copy occasionally, but it's just one copy and then the body goes back and makes a new one. When these become actually dangerous is not when it's in the mRNA copy, it's actually when it's in the true DNA strand, where the DNA strand has this mutation. So generally when we talk about these mutations, we're actually talking about the DNA, the actual code, not the mRNA that was made from it. But you can still illustrate it with this. Okay, and then it asks what are the causes of mutations. We call them mutagens. A mutagen is anything that causes a mutation in DNA. And they can be carcinogen, where they lead to cancer. They can be teratogens, where they lead to birth defects. Anything that mutates, the general term is mutagen, and it can be like UV radiation, like from the sunlight, chemicals, cigarette smoke, radiation, like actual radiation. So yeah, any of those things can cause mutations. Okay. Now, once they have finished, once you've finished your discussion of mutations, then it's their turn to apply it. And to apply it, they're going to build the myosin gene. And the myosin gene, it's the same process, but the one key difference is the myosin gene is on the bottom strand. So you might introduce this before you let them go and do it, right? The myosin gene is on the bottom strand. So if we always read the gene 5' to 3', we're going to be reading the myosin gene this way, this myosin gene. So when they write it down, so this says ATG right here, or TAC, their complementary base pairing, they're going to write on their little strip of paper, they're going to write it like this. So U, oh, I don't know what I'm doing here. What am I saying? Oh, and it has a Tata box up here and a polyadenylation signal down here. Okay, so it's got the same thing. But you're going to write it backwards, right? So if I see Tata box, I'm actually going to write it U-A-U-A-A-A-A-C-G-C. So even though I'm reading it this way, I'm writing it that way. Does that make sense? That just makes it easier when they go to translate it, right? They can do it however they want, but they need to realize that we're reading it this way, not that way. They can just turn the strand upside down, they can stand on their head and read it, whatever, but it's going the other way. And you need to emphasize that DNA genes, they're on both strands. They're not like on one strand or the other. They're on both strands, and in this case, this one's on the bottom strand. And they'll do the myosin gene, and then you can do the same thing. You can walk around. We've got a key. You walk around and compare the myosin gene and see how they did. And again, don't let them take them home. Take the beads off and put them back, or otherwise we're going to run out of beads. All right? Okay, so I think that's it for the lab part. Let me take a look at the homework. I'm not sure if I need to do something for the homework or not.